Good morning, dear students. Today we are going to study the first lesson in Unit 3. It's about the genetics and the hereditary traits. Our first lesson is the main principle of the hereditary traits. We are going to study how does we inherit our traits. But at first, we have to understand the objective of our lesson. By the end of the lesson, we are able to determine the difference between the hereditary and the acquired trait and explain why Mendel chose the pea plant for his experiment and identify Mendel's two laws of hereditary and identify the concept of the dominant and recessive traits and determine the dominant and recessive traits in Mendel's experiment for the pea plant and determine the dominant and recessive traits in the human beings. Also, we have to understand the difference between the hereditary traits and the acquired traits and understand the difference between dominant traits and recessive traits. Thousands of years ago, a man has observed that some traits are transmitted from one generation to another, and scientists have called that hereditary traits, such as or like the color of the hair, the color of the skin, the number of fingers, and blood groups. Some traits are not transmitted from one generation to another, as they are called acquired traits. So now we have two traits, hereditary traits, which transferred from a generation to another, and acquired traits, which gained by the human beings during its life. Mendel is the father of the genetic science. The experiment by Mendel placed for the basis for the scientific studies of the hereditary traits. This is according to the results reached by Mendel, and over the years, hereditary scientists have gathered a lot of information from the reasons why the living beings have the forms they have or behave why they do. How did Mendel start his experiments and what are the results he reached? Let's see a song about the Mendel experiment, then we will discuss the experiment itself.
There was an Austrian monk called Gregor. He looked at something never done before. I said Gregor Mendel was his name. He wondered why sometimes we looked the same. Using peas, he looked to their height. He worked all day and all night. The first he crossed was a tall and a dwarf one. Their offspring were all till F1. He was at no loss. He went for a second cross. He did it with two of these tall. What did he see? I said, what did he see? F2, one dwarf. The tall ones, there were three. Now, what was his conclusion? To him there was no confusion. Features came in hereditary units. This was his message. One was dominant and the other recessive. His work turned out to be quite prophetic. It was the start of modern genetics. Word to the brother. Peace.
Sadly, Mendel never got a chance to celebrate or be celebrated. His momentous discoveries were totally ignored in his lifetime, even though about 120 copies of the journal containing his paper were sent to libraries around the world. In contrast, Darwin, his contemporary and fellow giant of biology, won great fame within his. Perhaps if Darwin had heard about Mendel and his laws, much would have changed for both of them. It almost happened. Mendel mailed a copy of his paper to Darwin, but apparently Darwin never opened the letter. Mendel died in 1884 at age 62. It took another 16 years for the world to finally learn about him.
Oh, why, dear God, did you make it so complex to understand the offspring that result when there is sex? But there is one monk among us who can tell how it all works. We feel like jerks next to Gregor Mendel. The answer's in my garden where I've planted different peas and sprinkled pollen as I pleased and counted out the progeny. What did you discover in your garden with your peas about those factors we can't see but which explain our family trees? Here's the news, they come in twos, they separate, it's up to fate. If an egg or a sperm has a trait that will dominate. Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate. And when they join together, my forecasting's most impressive. Bet you 3 times out of 4 I'm right unless they're both recessive. To what do you owe your success? To counting and my green thumb. But where these hidden factors are, well, that I cannot fathom. Here's the news, they come in twos, they segregate, it's up to fate. If an egg or a sperm has a trait that will dominate. Later on the world was odd at what he learned from those pods. But back then no one hurrahed Gregor Mendel but his God. Back then no one hurrahed Gregor Mendel but his God.
After that 2 movies, I hope you understand what did Mendel do to establish the genetic science. He chose the pea plant to make his experiment. But the question now is why Mendel chose the pea plant to conduct his experiment for his research, and his choice of this plant was due to the following reasons. Number 1, it's easy to plant the pea plant and it grows fast, which makes easy to be distinguished. Number 2, the life cycle of the pea plant is short. Number 3, the flowers of the pea plant are hermaphrodite. It means that the flower is bisexual and can self-pollinate itself. Number 4, it can be easily artificially pollinated by man. Number 5, pea plant produces large numbers of plants in a generation. Number 6, which is the most important one, that there are several types of peas that have a pair of contrasting traits or different traits. We call that allelomorphic traits that can be identified easily. Some plants are long-stemmed and the other are short-stemmed. The flowers of some plants are white and the other are purple. The buds of the pea could be green in color or yellow as shown. Mendel chooses 4 different traits or 4 contrasting traits to observe or to distinguish in his experiment. The number 1, seed shape, that we have a round shape in one type, the other the wrinkled shape seed, and the seed color is yellow for the round one and the other is green. The bud shape is the smooth in the first type and the bud color is green, while the other type is pinched or sinuous and the bud is yellow. The flower color is purple in the first type while it's white in the other type. The flower position at the side of the stem, while in the other type it's at the end. So the last trait is the tall stem, which characterizes the first type, and the short stem for the other type. There is another video to discuss how did Mendel do his experiment.
In the early 19th century, biologists believed that blending was responsible for pink offspring when you crossed red and white snapdragons. We now understand that this is not the case for the traits that Mendel studied. But how did he figure that out?
The reproductive structures of the pea plant are contained in each flower on the plant. They are the stamens and the pistil. A flower self-pollinates when pollen from the stamen is transferred by wind or insects to the sticky surface on the pistil. A pollen tube develops and grows down the pistil to the egg cell in the ovary. The sperm nucleus in the pollen tube combines with and fertilizes the egg cell. Mendel had to ensure that he could control the parentage of seeds. He wrapped plants that he wanted to self-fertilize so that they wouldn't be pollinated and fertilized by other plants. He clipped off the anthers of flowers on other plants so he could pollinate the plants himself with the pollen from other specific plants. This is cross-pollination. It results in the fertilization of the egg cell of one plant by the sperm nucleus from another plant.
I must make this clear for the Society of Natural History. These plants all had terminal flowers, flowers that grow from the ends of the stem. After they had been self-fertilized, they produced plants that had terminal flowers. I called these plants my P1 generation, parent plants that I knew were true breeding. These plants produce flowers that grow from the side of the stem, axial flowers. After these plants had been self-fertilized, they all produced plants that had axial flowers. These plants are true breeding. I decided to make my first cross with true breeding plants. Cross-fertilized true breeding plants for terminal flower with true breeding plants for axial flowers. All the flowers produced in the F1 or first filial generation had axial flowers. I wondered where the terminal factor went. I decided to cross the F1 plants. The F1 plants were not true breeding. That is, they did not produce offspring like themselves. Of the plants of the F2 or second filial generation, 3/4 produced axial flowers and 1/4 produced terminal flowers. The factor for terminal flowers had come back. Curious to know more, I let each plant of the F2 generation self-fertilize. Of the plants produced, 1/4 were true breeding for axial flowers, 1/2 were not true breeding, or hybrid, but produced axial flowers, and 1/4 were true breeding for terminal flowers. Clearly, the hybrid plants contained factors for both characteristics, but produced only axial flowers. The factor for terminal flowers had not disappeared. It was only masked. When it combined with a plant that had a similar factor, it produced offspring that had terminal flowers. To explain these results, I believe that the hereditary factors must occur in pairs. These factors must separate in the parent plant, and then each parent must give one factor randomly to the next generation. I will call these factors either dominant or recessive. The dominant factor masks the recessive factor. In this case, the true breeding axial plants have two dominant factors, capital A, capital A. The true breeding terminal plants have two recessive factors, small a, small a, so the trait can show. The hybrid plant has one dominant factor, capital A, and one recessive factor, small a, but only the dominant factor, axial flower, shows.
To understand the genetic Genetics is to study the hereditary traits and how the traits are passed from parent to offspring. There is a picture to show you when the cross-pollination takes place between two living organisms of different traits, like the white dog and the black dog. We have very alternative choices for the childs of the offsprings to be produced. The study of the hereditary traits with the work of Gregor Mendel and his pea plant garden. Mendel was an Australian monk that lived in the mid of 18s. Mendel noted that the size of the pea plant varied. He crossbred these pea plants to find some surprising results. Mendel cross between tall pea plants yielded all tall pea plants. His cross between small pea plants yielded all small pea plants at the first. Mendel's cross between tall pea plant and the small pea plant, as you saw in the last video, and the yield was all tall pea plants. Mendel then crossed this second generation to all pea plants and ended his 1 out 4 being small. Genetics, according to that results, there are 3 basic kinds of genes. There is a dominant gene or dominant trait, which is the gene that is always expressed and hide others. Recessive gene, that is the gene that's only expressed when a dominant gene isn't present. And the co-dominant genes or hybrid genes, the genes that work together to produce a third trait. Dominant and recessive genes. A dominant gene will always masks a recessive gene. And then the picture, the Windows beak is a dominant. Not having a Windows beak is a recessive gene or trait. If one parent contributes a gene for a Windows beak and the other parent doesn't, the offspring will have a Windows beak. Punnett square, a tool we use for predicting the trait of an offspring. Letters are used as a symbol to designate genes. Capital letters are used for dominant genes always. Lowercase letters are used for recessive genes. Genes always exist in pairs to express a trait. A Windows beak dominant would be symbolized with a capital W, while non-Windows beak recessive would be symbolized with a lowercase w. Father, as in picture, with no Windows beak, and mother has a Windows beak. All organisms have 2 copies of each gene. One contributed the father and the other contributed by the mother. A homozygous pair, when 2 copies of the same gene, it means that we have a pure trait. Heterozygous, when 2 different genes aggregate, this is called hybrid dominant. For the Windows beak, when we get WW letters, capital, has a Windows beak, it means that a pure dominant or homozygous dominant. When a letter capital W and lowercase w, we has a Windows beak, but with a heterozygous or hybrid dominant. When 2 lowercase w's, there is no Windows beak. This is an homozygous recessive or pure recessive. Since Herman has no Windows beak, he must be 2 w's with lowercase. Since the other one has a Windows beak, she could be either two W's with capital letter or with one capital and lowercase. Eventually, WW lowercase is a homozygous recessive or pure recessive. Either capital W and lowercase w, it means a heterozygous or hybrid dominant. When there is two W's capital, it means that we have a dominant hybrid, a pure dominant or homozygous dominant. Genetics, we have also, we can use a Punnett square to determine what pairs of genes we have. As you see that we have all the probabilities, letter capital W and small for the heterozygous or the hybrid dominant, and we have two lower cases W for the pure recessive. When we calculate the probabilities, we have at the first square capital W and small. This is a heterozygous dominant or hybrid dominant. The other probability for small and small w lower cases, it means we have a pure recessive trait. The other probability will give us a capital W and small letter lowercase, which determines that we have a hybrid dominant, and the other probability will be as shown before two lower cases w, which means that we have a pure recessive trait. Now let us understand the principle of complete dominance done by Mendel. Mendel deduced from the previous experiment we know some important assumptions. Number one, the color of the seed depends on factors. These factors present in plants transmitted from one generation to another by means of gametes. So gametes responsible for the transmission of the factors of the genes from parent to offspring. There is a factor that determines the yellow color of the seed, and the other factor determines the green color of the seed. When these factors meet in the first generation, we call it aggregate. The yellow color factor is the dominant one over the green color factor, that is recessive, and this leads to the production of only yellow seed in the first generation, indicating that the yellow is a dominant over the recessive green. When the gametes in the first generation are produced by meiosis, these factors separate or segregate from each other, that they meet again to form the second generation. According to that, the assumptions done by Mendel, that he stated that there is hereditary traits are transmitted from parent to offspring by means of hereditary factors, which are known as genes. So now we understand that hereditary factors is called genes. A living organism, every hereditary factor trait is controlled by two hereditary factors or two genes, one from father and the other from mother. And these factors are similar if the traits are pure, and not similar or different if the traits are impure. And the living organism carries an impure trait is called hybrid. The two hereditary factors in every trait separates or segregate when the gametes are formed, where the gametes carries one factor to each hereditary trait. And Mendel has summarized the previous assumptions in law known as Mendel's first law or law of segregation, which states that the hereditary traits in a living being is represented by two hereditary factors, separate or segregated from each other when the gametes are formed, where gametes crossing carries one hereditary factor to each trait. We will show you another video to understand how to calculate the probabilities of the genetics according to Mendel's first law. Listen.
Hi, it's Mr. Anderson, and today I'm going to give you a beginner's guide to Punnett squares. There are a few mistakes that students always make when they're doing Punnett squares, so I hope to kind of clear those up. First of all, we should talk about the namesake. This is Reginald Punnett. He didn't really work with Punnett squares, however, he did work with genetics quite a bit. He did work with mimicry in the butterflies, and so his name is probably associated with genetics just as much as Mendel. And it's through the use of the Punnett square. Now, the Punnett square is often overused as just a quick way to solve genetics problems without really understanding what's going on with the genetics. And so I want to kind of get to that root. And if you could remember one thing from this whole video podcast, it's this right here. The two sides of a Punnett square represent the alternatives after meiosis. In other words, you have a bunch of genes and you give half of those genes to a sperm or an egg, and that happens through meiosis. And so the organization of those gametes, this case it's just a monohybrid cross, are going to be on either side of this, just like a flip of the coin. This would be for one parent, and then this would be the other parent on the other side. And so what do the boxes on a Punnett square stand for? They simply stand for all the alternatives that could occur if we had mating between each of these different gametes. And so let's get to some examples, and hopefully that'll help. So we're going to start with a monohybrid cross. A monohybrid cross is simply a cross that is looking at one trait. And so let's do one that's really, really simple. And so let's say we're crossing purple flowers that are homozygous purple with those that are homozygous white flowers. In other words, this is the dominant trait, this is the recessive trait. And so if you look at the parents, what you want to do first of all is figure out what are the possible gametes that could be produced in meiosis. In other words, this one could either give a big P or it could give a big P. What does that mean? It can only give one thing. It can only give a big P, or that dominant allele. And so if you're doing a problem like this, you don't even need a big Punnett square. One parent can only contribute big P. So let's look at the other parent. The other parent can either give a little p, and I try to make them really small, or a little p, because p's look the same. And so the other parent can only give a little p, and so we could put that on the other side. And so what are the opportunities that we could have as far as fertilization goes? Well, this one's automatically going to give a big P. The other one's going to give a little p. And so this is the only possible outcome we could get between a cross of a homozygous dominant and a homozygous recessive. And so you don't really need a big Punnett square. Now you could do that. You could fill it in, big P over here, little p over here. But if you do that, you're going to get the same thing in all of the boxes. And so it's still a 1-to-1 ratio. In other words, 100% of the time you're going to have that. Okay, so let's get to one that's a little more complicated. Let's say we have a heterozygous cross. And so this is for purple flowers as well. Well, if you look at this one now, the problem's changed a little bit. This one could give a big P, but it also could give a little p. And so we have to show both of those possible gametes of meiosis. And so this would be the big P, and then this would be the little p over here. So half of the time it's going to give the big P, half of the time it's going to give the little p. If we look at the other parent, same thing. It's going to give the big P half of the time, and it's going to give the little p half of the time as well. So we're going to put that here. Now we simply fill in the boxes. And so this would be a big P with a big P, because I'm taking this here and that there. This is going to go over to here to give us a big P and a little p. By convention, we usually write the dominant allele first. So that would be one alternative here. Here would be a big P, little p as well. So we get the big P here and the little p here. And then finally we're going to get little p over here and a little p over here, since they're each contributing a little p. And so what do we get from this cross? Well, we get a 1 to 2, since these are exactly the same, to 1 genotypic ratio, because the genotype is the letter. So there's 1 that's like this, there's 2 that look like this, and there's 1 that looks like that. So that's going to be our 1 to 2 to 1 genotypic ratio. What about phenotypic? Well, this one's going to be a purple flower, and so are these other 2. And so if we're looking at the phenotypic ratio now is going to be a 3 to 1 ratio. We're going to have 3 purple to every 1 white that we have. Okay, let's try another one. Let's say we're looking at incomplete dominance. So incomplete dominance, a snapdragon would be an example of that. A snapdragon has 2 genes. If it has a red gene and a white gene, then it's going to be pink. And so this one actually has 2 alleles that it can contribute, and the same on the other side. And so we just write those out. So this would be the parent. This would be a 50% chance of giving the red, 50% chance of the white. And then the same thing on this side over here. So now if we fill in our Punnett square, like that, what do we get for all the different choices? Well, now we have a 1 to 2 to 1 genotypic ratio, but we also have a 1 to 2 to 1 phenotypic ratio. So if you're doing a question where it's incomplete dominance, you use a Punnett square the same way. Codominance would be the same way. The difference is in incomplete dominance, the heterozygous, or the hybrids, are going to be somewhere between the two. If it's codominance, they're going to express both of those genes, both of those proteins. And now let's try one that's a sex-linked chromosome, or an X-linked chromosome. And so in this one, we've got a parent. So this is a mom, because she's XX chromosome, and she's a carrier of, let's say, color blindness, the gene. And this is a dad that's normal. And so I'm going to put dad up here, XY, because half of the time he's going to give the X and half of the time he's going to give the Y. If we put mom over here, I'm going to put that carrier up there. She is not colorblind because she has one deficient gene, colorblind gene, but she has another gene that works well on her other X chromosome. So if I fill in this one here, it's going to be XCX. So this would be a female because 2 X chromosomes, but they're going to be a carrier of that gene. If we look down here, this would just be a normal female. If we look down here, I'm grabbing the X from here and the Y from up there, so that'd be XY, so that'd be a normal male. And then if we look at this one right here, this would be a male who's colorblind. The reason he's colorblind is that he doesn't have an X chromosome or another gene as a backup copy to that. So those are monohybrid crosses, and usually students do fairly well on those. Next are dihybrid crosses, and this is where the mistake really starts. And so if we look at this parent, this is a typical dihybrid cross. Let me tell you what the letters stand for. The R stands for round pea seeds, and the Y stands for yellow seeds. If it's the recessive, that stands for wrinkled, and if it's a little y, that stands for green. And so we're looking at a dihybrid, so that means 2 traits. We're looking at seed shape, round or wrinkled, and seed color, yellow or green. So now we have to do a dihybrid cross. And so the tendency, if we look at this parent, the tendency is to see that there are 4 letters over here, there's 4 boxes over here, and then you just simply write them out. Big R, big Y, little y, and then you get a weird answer and you don't know what to do with it, okay? That's wrong. That's a mistake, okay? Whenever you're figuring out the gametes, remember that you have to give 1 of each letter. In other words, each of the gametes is going to have 1 of each of the alleles. And so let me clear this mess out of the way. So what do we do? Let's say this parent right here. And I'm going to write it up here so it makes a little more sense. Big R, big Y, little y. What possibilities could they produce? So they're giving 1 of each letter, remember. Well, they could give the big R and the big Y. So big R, big Y. That'd be one possibility. They could also give the big R and the little y. So they could give the big R, little y. They could also give the little r, big Y, or the little r, little y. Since they're giving 1 of each color, there's only 4, 1 of each letter, there's only 4 possibilities that they can give. So it's getting to be kind of a mess, but those are the 4 right here. And so those 4 are going to go across the top. So we got big R, big Y, big R, little y, little r, big Y, little r, little y. So those are the 4 gametes that you could produce. In other words, with this parent, you can only get 4 combinations of each of the 2 letters. Same thing down this side. So it's going to be same thing on written down this side because this other parent is going to be exactly the same. Okay, so it would take me a long time to write all of those out. So let me throw those in here. So these would be the parents, all the possible gametes you could get. And if I fill those in, these first 9, if you look at them, let me get a color. It's different. Let me grab a yellow color. And so this one right here is going to be round and it's going to be yellow. So it's going to be round and yellow. And this one you can see it's going to be round and yellow and round and yellow and round and yellow. And yellow and round and yellow and round and yellow and round and yellow and round and yellow. In other words, 9 of them are going to be round and yellow in shape. And the only reason why is that the R is dominant, and so you could have one like this where they're hybrid for both and they're still going to be round and yellow. And so let me try to add the next ones. So what about these next ones? Well, the next ones are going to be round and green. So let me get a different color. So these ones are going to be round and green because the round is dominant, but they don't have the dominant for the yellow, so they're not going to be. Let me get rid of that. Let's add the next ones. So these ones are going to be wrinkled and yellow. So again, I got to get a different pen. So these ones are going to be wrinkled and yellow. And then if we look at the last one, it's going to get all of the recessive alleles. And so that one's going to be getting green. It's going to be green and wrinkled. Okay. And so when we say there's a 9 to 3 to 3 to 1 ratio, that's just a typical dihybrid cross. We're going to have 9 of the phenotypes that are round and yellow, 3 of the phenotypes that are round and green, 3 of the phenotypes that are yellow and wrinkled, and then only 1 of the ones that's not. And that's only going to work if you are able to set up your gametes correctly on the side. Now it's super hard for you to answer a question like this on a test. You're rarely going to have to draw a dihybrid cross, but it's important that you understand the concept of it because most of the genes inside your body are not caused by one gene, they're caused by multiple genes. So how tall you are is caused by probably a dozen different genes inside your body. So you can imagine how big the Punnett square is going to be for that. An example of that I'll leave you with would be this one. So let's say we have a parent here, and the parent is big R, big Y, little y, little r, little y, little y. The question I'm asking you is, how big would your Punnett square have to be? And so you're going to have to figure out what are all the possible gametes that you could get from both of those, and then draw it out, figure out all the possibilities you can get. If you're thinking it's going to be a 4 by 4, you're doing way too much work. And so those are Punnett squares, and I hope that's helpful.
Now, according to the last video, we understand that Mendel's do another experiment to understand what would happen if he cross-pollinate 2 different pair of traits. He obtained the ratio 9 to 3 to 3 to 1. So he made his second law, which is called Mendel's second law. In that law, which said that and Mendel assortment of the hereditary factors. In that law, he stated that if 2 individuals that are different in a pair or more of alternative traits or contrasting traits copulates as shown in the last figure, the traits of each pair is inherited independently and appears in the second generation at a ratio 3 to 1. Now we study 2 of Mendel's law. Mendel's first law, which is called law of segregation of factors, and the Mendel's second law, which is called the law of independent assortment of the hereditary traits. And now, what about the dominant and the recessive traits in the human beings? A number of human hereditary traits follow the Mendelian hereditary, where the traits is controlled by one pair of genes. It can be dominant or recessive. The individuals who receive the least one dominant gene from either parent will have the dominant trait, and those who receive a recessive gene from both parents, that recessive traits will appear in them. Observe the following figures to identify some of the traits that follow complete dominant principle in the human beings. The ability of rolling tongue is one of the dominant in the human beings over the inability of rolling tongue. Also, the separate earlobe dominant, that connected earlobe trait. We have also the wide eye traits, which is dominant over the narrow eye traits, and the curly hair traits dominant over the straight hair trait. Also, we have the presence of dimples in the face is dominant trait over the non-dimpless trait. We have the non-presence or absence of freckles in the face trait, which is dominant trait over the presence of the freckles trait. And now, let's check our information about the lesson. Question number 1. Mention the scientific term for each. Letter A. A science that researches the transmission of the hereditary traits from one generation to another by studying the similarities and difference between the parent and offspring. This is our lesson. It's called genetics. Letter B. The characters ready to be transmitted from one generation to another. This is what we inherit from our parents, so we called it hereditary traits. Letter C. The trait that appears in all individuals of the first generation in Mendel's experiment. This is the strong gene or the strong trait. We call the strong trait as dominant trait. Letter D. The appearance of the hereditary trait in the individuals of the first generation when two individuals copulate and one of them is carrying a pure hereditary trait contrasting the trait carried by the others. This is the statement which explains Mendel's first law. And now, I hope you understand what we study in our first law in genetics about Mendel and his experiment and his two laws. We will see you again in our second lesson.